Eh bien, merci à notre tour de nous avoir éclairé sur une dimension que nous, cliniciens, on a peut-être tendance de temps en temps à négliger, tous les aspects anthropologiques de ces problèmes de santé. Je vais immédiatement passer la parole au professeur Kevin Ariën, professeur de virologie à l'Institut de médecine tropicale, qui va nous entretenir des aspects virologiques et de diagnostic de laboratoire. Je vous en prie.
Bonjour tout le monde, merci pour l'invitation et je suis content de parler ici devant vous aujourd'hui, mais mon français n'est pas assez bien, donc je vais faire mon parole en anglais, excusez-moi. So, I will talk to you about what we currently know about Ebola virus and about diagnostics. I could talk about HIV as well, but I'll do that another time. So, this is basically one of the old images that everybody knows. So Ebola is an enveloped virus, and it has a negative stranded RNA genome. Many viruses have these RNA genomes, and it has this typical filamentous shape. It is endemic in Central Africa since '76, since the discovery, and ITM, as we heard by Dr. Piot previously, has been pivotal in that in '76, together with Dr. Piot and Professor Patin and Dr. van der Groen at that time. So it causes severe hemorrhagic fevers in humans, in non-human primates, but also in a number of other animals, and I guess we'll hear more about that in the other talks. And it has, of course, high case fatality rates. Ebola virus belongs to the family of the filoviruses, and there's different members in these filoviruses. I don't know how this pointer works. So there's Marburg virus, Lovio virus, which was isolated from bats in Spain in 2003, and then a number of Ebola types, 5 types actually, with different case fatality rates. And the most deadly form of Ebola virus is the Ebola virus, Zaire, the one that is currently also causing the epidemic in West Africa. The second most lethal Ebola strain is the Sudan virus, with slightly lower case fatality rates, but still around 50%. A more recently identified Bundibugyo strain, identified in 2007, with a lower case fatality rate of around 25%, but there have only been 2 outbreaks until now, so we don't know too much about that species of Ebola yet. And then there's two separate or different cases, that the Thai forest, or also known as Côte d'Ivoire strain. There was only 1 case, the person survived. And then there's the Reston virus, isolated in the US, in a place Reston, which is non-pathogenic to humans. Humans get infected, but they don't develop any disease. Filoviruses are actually old viruses. So this is phylogenetic tree using complete genome sequences from previous outbreaks. So there weren't that many sequences, full genome sequences, known for Ebola. We know many more now, so this tree might change or be updated. But if you look at the dating, this is dating of the most recent common ancestor. The most recent common ancestor of these filoviruses dates back about 10,000 years. So these viruses have been around for quite a long time. This is more recent data with including full genome sequences from Sierra Leone and Guinea. And Stephen Zaïre et al., they looked at how different this virus that currently circulates in West Africa is from previous outbreaks with the same type of virus, with the Ebola Zaire strain. And apparently it dates back about 10 years, about a decade. So the virus has evolved over about a decade to cause, and now causes this outbreak in West Africa. So where does it actually originate from? As was pointed out before, besides this 1 case in Côte d'Ivoire, all cases occurred in Central Africa, and there's a few hotspots of outbreaks in DRC, of course, also the border of the Republic of Congo and Gabon. There have been outbreaks, the southern part of Sudan and Uganda. So where does this virus come from? It's a zoonosis. Viral RNA and Ebola antibodies can be found in African fruit bats, and 3 types of fruit bats, and I won't even try to pronounce these names. The interesting thing is that in contrast to the Marburg virus, which was actually isolated from bats, Ebola has never been isolated from these naturally infected animals. So that's really an interesting thing, and something we have to further do research on. We also know that bats are very numerous in equatorial Africa. In many parts of Africa, they're also hunted for food in many of these places. Ebola is lethal for non-human primates, for duikers, that's a kind of antelope species, and also for bush pigs or wild boars. And all of these animals are also hunted and consumed as bushmeat. But probably the picture is bigger. The ecology is the ecology is larger than that, and there's maybe room for an intermediate host. And pigs might be an important reservoir there. And I won't tell too much about that because I think we'll hear more about that in the later talks. But there is experimental evidence with infection of pigs with Ebola Zaire. And also the Reston virus has been found in the Philippines in pigs. Transmission, most of you know, of course, there is close contact or direct contact with infected patients or cadavers are required. Most infectious bodily fluids are blood, vomit, other excreta, genital and nasal secretions, sweat, urine. We also know that sperm contains virus and can be a reservoir that remains positive for Ebola virus quite a long time. And then direct contact with mucosal surfaces or skin abrasions can cause these infections. Okay, looking at the current outbreak, and I think much of that was already introduced. So it started late December 2013, but it was only recognized in March and even later towards the summer of 2014. So a lot of time has been lost there. So the epicenter of the epidemic was actually here in Guinea, Kwakidu and Macenta districts, and then just south of Macenta and Kwakidu, Lofa district in Liberia, and then Kailahun and Kenema district in Sierra Leone. So this was for the first time that we had a multi-country outbreak. There was one single case of Ebola virus infection in Côte d'Ivoire. But this epidemic is different because this is an area that is much more densely populated than Central Africa. It has large cities, and these large cities, the virus has been found in these large cities. And it's also part of Africa with lots of mobility and trade going on across these borders. So that actually helped in the spreading of this virus. So if we look at the current situation, over 20,000 people got infected, almost 10,000 died of it. Majority of the infections occurred in Sierra Leone and Liberia, a bit less in Guinea. It's by far the largest outbreak of Ebola virus disease until now. But in terms of the incubation period and the duration of illness, also the case fatality rates and the reproductive rate, there's nothing really special about this outbreak. So it's all within the ranges that we've seen with previous epidemics. Approximately 800 of these healthcare workers have become infected, and almost 500 of them have died. So that was also mentioned before. So is this really unexpected, this outbreak in West Africa? If we look at the distribution of these different bat species, the 3 bat species that are known to carry the virus, or at least the RNA of these viruses, they're supposed to be the reservoirs. So they live, or their distribution is in Central Africa, that's for clear, but also these coasts of West Africa. So the potential reservoir is there. So it's probably not that surprising. If you look at the countries at potential risk of Ebola virus infection, also these countries in West Africa are regarded as, or predicted as okay. So The epidemic is currently slowing. That's what we see from, or hear from the latest numbers that we receive from WHO and other organizations. And this is a figure in a New England Journal of Medicine publication in 2014, end of 2014, where they actually predicted how the epidemic would evolve. And if you look at the actual numbers, this is the numbers from the latest WHO report of this week, last week, you actually see a drop in the numbers of new cases, especially in Liberia and Sierra Leone. Also in Guinea, although there was a small increase again in Guinea the week before that. Okay, so I wanted to come to this part actually, to some new developments, and of course there's developments in terms of vaccines, of new antivirals, blood products, plasma or blood donation, and also diagnostics. And for the sake of time, I will only discuss, or will only focus on diagnostics, and I'll have to say something very shortly about vaccines as well. So for diagnostics, the differential diagnosis is very important, especially in West Africa, where malaria and Lassa is also endemic. The current standard for diagnosis is reverse transcriptase PCR, so detection of the viral RNA. And there's also antigen detection ELISAs available. Also antibody detection, so serology can be done, but it's regarded as being less relevant for lab diagnosis. Many of these patients actually don't develop good antibody responses. So if you look at the evolution of the viral load, and this is days post-onset of symptoms, so you could already detect viral RNA in the early phase of the infection. However, with antigen, it's the same. You can detect the antigen, but the curve is shifted slightly to the right. So the earliest detection and actually best detection is real-time PCR, and there's also reverse transcriptase PCR, and these are also very sensitive assays. If you look further at the course of the disease, this is already about 10 days into the infection, so where the plasma viral load is dropping. You see that the antibody titers are being built up, but, and this is interesting, the virus can still be detected, even if it's undetectable in the blood. It could still be detected in other body fluids like sweat, like urine, and we also know that semen is a reservoir that remains positive for a long time. Okay, so this is the algorithm for laboratory testing. And since October last year, the ITM is responsible for the diagnostic testing in Belgium as a reference center. So as I said, the differential diagnosis is important. A blood specimen then of a suspected case is sent to a reference lab, either BSL-3 or BSL-4. In Antwerp, we have BSL-3 level, actually BSL-3 plus level. I come back to that later on. So if the PCR test is negative, another sample is taken within 48 hours, and that's up to the decision of the medical team to retest yes or no. In most cases, this is done. If the PCR or if the test is positive, we do a second confirmation test. We do that in-house immediately, but the sample is also sent to a reference center, an international reference center in Hamburg, Bernard Nocht Institute for Tropical Medicine, which try to isolate the virus and then confirm by sequencing. Okay, so the Ebola virus diagnostics at ITM, how do we do it? We have a well-equipped biosafety level 3 facility. So we're doing these diagnostics for the Belgium territory since October last year. And we have a dedicated room within our biosafety level 3 lab, which we upgraded to biosafety level 3 plus. And what does that actually mean? So we have further restricted access there, and only 6 highly trained people can enter this laboratory. And these are 2 of our colleagues in action, doing the neutralization and preparing the sample for viral RNA extraction. We do the malaria rapid tests in the BSL-3 lab as well. We extract DNA from whole blood to do plasmodium PCR, and also do viral RNA extraction on the plasma, because that has the highest sensitivity, to do then reverse transcriptase PCR, targeting 2 different genes. We target the glycoprotein gene, that's an in-house PCR, and an L gene PCR, which is commercially available. Okay, something about, very briefly about Ebola vaccines. There's 3 vaccines that are currently in development, or at least furthest in development. And these are actually the 2 that are now being, or are up for trial, hopefully, in West Africa. And there's this recombinant vesicular stomatitis virus vaccine, which was which was designed by the Canadian Public Health Agency, and then sold off to Newlink Genetics, and also Merck is now involved in the further development. So this is a live attenuated vaccine, a vesicular stomatitis virus vaccine vector, where the glycoprotein of the VSV virus was replaced by Ebola glycoprotein. This GSK vaccine is a chimpadenovirus vaccine, so a common cold vaccine, where E1 was replaced by Ebola virus glycoprotein, and E4 was deleted. So this is just a vector vaccine, it's not a live attenuated vaccine. Why do I do this introduction? Because I wanted to tell you about an interesting case that we had here in Belgium in December last year. We had a medical doctor returning from an MSF treatment center in Liberia, Monrovia, after a needle stick injury. And she was hospitalized at the Saint-Pierre Hospital here in Brussels. And 36 hours after the needle stick injury, she received this recombinant VSV glycoprotein vaccine as post-exposure prophylaxis. There's only one single case described before of a German lab worker, BSL-4 lab worker, in Hamburg, who received this same treatment. So this is actually the second case. The person got vaccinated, developed fever, which is not uncommon with live attenuated vaccines. And then we received samples just prior to the vaccination and at several time points after the vaccination. And the interesting thing is that in the L gene PCR, the sample remains negative, but in the in-house glycoprotein PCR, the sample was, or the patient was 5 consecutive days positive, scored positive in the Ebola testing. So we just published this last week in Clinical Infectious Diseases to re-emphasize the importance of doing a double target PCR, because this is sometimes forgotten, and especially with the vaccine developments ongoing. So the current 3 vaccines, as I said, are all using the glycoprotein or based on the glycoprotein, but glycoprotein is, as I said, we use an in-house PCR, so it's used for diagnostics, but there's also antigen-based tests looking at or detecting glycoprotein. And even if you look at other investigational vaccine targets, many other genes like nucleoprotein and VP35, VP40, and so on have been put forward in literature as vaccine targets. And there are also different diagnostic tests have been described that target these genes. Okay, Ebola rapid diagnostic tests, what's going on there? So why do we need them? Of course, they're important to rapidly confirm suspected cases, to help in the triage early on and in making these clinical decisions. They could also minimize the window of transmission, and they could be very helpful in contact tracing. Of course, they need to be rapid, and rapid is actually within 5 to 15 minutes. WHO puts the limit at about 30 minutes, half an hour. They need to be sensitive, and many of the antigen-based tests have issues with sensitivity, especially compared with the standard PCR tests. And of course, they should also be safe and easy to use. Another delicate point, especially when using these dipstick tests or lateral flow test devices. So there's a couple of tests that have received emergency use authorization by FDA, like this BioFire test. This is also PCR-based test. It takes about an hour. Our test in Antwerp runs about 3 to 4 hours. So that's a time reduction there. And then also emergency use authorization by WHO just recently, yesterday or the day before, for this Corgenix test. It's an antigen-based rapid test. Just the last slide now. Also at ITM, we're working on new diagnostics together with a company called Biocartis. That's a company from Rudy Pauwels. And he has this interesting platform, the IDILA platform, which was launched last year in September. And this machine actually combines the RNA extraction on whole blood and the qPCR detection in about 90 minutes. And we're sure actually that we can further reduce that time to around 60 or 70 minutes. But the advantage is here that you could just add, you don't need much manipulations, it's 1 or 2 minutes of hands-on time. You add a drop of blood into this cartridge, you close the cartridge, you put it on the machine, and everything happens automatically. So we're working on that, and we're also trying to bring this to the field to West Africa. I would like to stop here. These are the people I would like to thank because they have been very inspiring for this talk. Thank you.